Next in line is Zohar Arnon. So, Q here. All right, yes? All right, so we got, okay, we can upload and
איפה זה היום? זה כאן פה, רגע. מה קרה לך ברגל?
All right, time is ticking.
Okay, hi everyone. I'm Moran from Lee Adler Abramovich Group. I will present part of my PhD work on improving the mechanical rigidity of hyaluronic acid by integration of supramolecular peptide matrix. So hyaluronic acid is widely used in the cosmetic and medical field. However, its poor mechanical features and fast in vivo degradation hinder its utilization. One way to strengthen HA is to cross-link it, and we provide a different approach of reinforcing the HA with the FMOC diphenylalanine peptide. So we combine these two, we mix these two compounds, HA and FMOC-FF, together to form a homogeneous fibrous network, transparent and injectable composite hydrogels. These hydrogels represent notable mechanical features with improved stability to enzymatic degradation. In vitro viability test with few types of cells show very good biocompatibility features. And another interesting experiment that we did, we harvested limb buds from a chick embryo, embedded them in our hydrogels, and placed them together on the upper membrane of a fertilized egg. The results were amazing. The limb buds developed into fully, I'm not sure if you see it clearly, but into fully leg or wing formation after 7 days in ovo. So based on our results, which was recently published, we see the great potential of these hydrogels to serve as 3D scaffolds for drug release and tissue engineering applications. Thank you. Poster number 21. Thank you.
Thank you, Moran. Next is Zohar. Tom, sit in the chair. And Zohar, coming from the Gazit group, will tell us about structural manipulation of self-assembled supermolecular characterization applications. Awesome, yeah.
All right, so hi everyone. My name is Zohar, a PhD student from Ehud Gazit's group. And I'll tell you about my project called Structural Manipulation of Self-Assembled Supramolecular Polymers. So first, what is peptide self-assembly? So in this short video, we can see someone went through a lot of effort to organize these magnets stably. But if we give this system a nudge, the system spontaneously self-assembled into this elongated structures. So we do basically the same, but instead of magnets, we use diphenylalanine, which is this molecule. And given the right conditions, it forms elongated structures like this. So we call these nanotubes because we can actually see the cavity. So what is this good for? There are many unique properties for these assemblies, and the potential is very diverse, from reinforcements to drug delivery and energy storage. So what we did is we took the microfluidic platform, you can see there in blue, and we injected the assemblies into the device, and then we could inject whatever solution we want and see how the solution affects the assembly. And this is all the same nanotube throughout time, from left to right, at different conditions, so we can monitor the growth and shortening of the assemblies and see how different solution affects the growth. I show some other projects in my poster, so if this interests you, just come see me at poster 22, And thank you.
Thank you, Zohar. Next, coming also from Professor Ehud Gazit, is Tom Gutman, talking about light-controlled 3D nanowires, self-assembled nanowires. Ella, come to the chair.
Okay. Hi everyone. I'm Tom. I'm also from Ehud Gazit's lab. My goal is to mimic bacterial nanofibers that are called pili using self-assembling peptides. So there's a very interesting type of bacteria. Let's call it GS. And this bacteria expresses very thin fibers on its outer surface. They're called pili. And these pili are proteinaceous, and they interact with metal oxide particles and metal ions that are found in the soil where these bacteria usually grow. So there's an interesting biological story behind this, but what we're doing is we're looking at the amino acid sequence of its monomer, the monomer of these fibers, and we derive short peptides from it. So there's, for example, here a 20 amino acid long peptide derived from two distinctly different regions of this protein that's self-assembled into nanofibers that are similar to the native fibers in certain aspects. And one thing we did with these fibers, inspired by the native system, is interacting them with metal oxide particles, where they co-incubate and become decorated by these particles, or interact them with gold ions, which become reduced into nanoparticles, and they decorate the fibers to very high extents. This can be put in different substrates and then can be used as a catalytic coating, reusable catalytic coating, or as self-supporting immersible films that can form a good scaffold for heart cells or static charge detector. And these things are not just illustrations, as you can see here again. Peptides, self-assembled nanofibers, metal decoration, and the different applications of this novel bio-inspired material. So with that, I would like to thank you, and please visit me at poster number 23.
Thank you, Tom. Next, we have Ella Bromberg from Fernando Patolsky's group, telling us about separation, desalting, peak concentrations, and controlled.
Hi, my name is Ella, and I'll be talking about light control, selective and collection and release of biomolecules. Basically, when you're sick, you want to be analyzed and then get a diagnosis and treatment that is very suitable for you. The problem is the information lies within biosamples, which are very complex. They contain different concentrations of various molecules, and it's very hard to handle. Usually it takes a lot of steps and a lot of methods just to get simple information from the samples. What we aim to do is create a filter that is based on nanostructures of silicon and is easily integrated into a FET device that could analyze the sample. So, on the left we have one pillar as an example. We modify the surface with an antibody, and this, when introduced to a biosample, is selective to a specific protein. When we activate the surface that contains a photoacid using UV light, we lower the antibody protein affinity and cause the controlled release of the protein. So we can later on, by microfluidics, introduce the protein to a FET device in a pre-concentrated and very purified manner, and easily analyze the sample. If you're interested, please come to poster number 24 and I'll explain some more. Thank you.
Thank you, Ella. Next, we have Itai Silver from Yoram Dagan's group, telling us about quantum spin liquid.
Give me a second. Okay, hello everybody. I'm Itai from Yoram Dagan's group. First one from physics, so this will be a bit different from what we've seen so far. Let's get into it. Imagine a material that has some magnetic moment inside, usually attributed them to electron spins. Naively, we would assume that if we lower temperature enough, those spins will order themselves in some pattern, like a crystal. However, given a material that has some correlation that results in geometrical frustrations, all those spins cannot be simultaneously aligned the way they would like to. In that case, there is a theoretical state of matter called a quantum spin liquid, in which even at no temperature at all, the spins continue to fluctuate like a liquid. And my quest is to find a material that does exactly that. So I worked with a very nice material, tantalum sulfide. It orders in a 2D layer of those little stars of David. And I made a simple measurement of the heat capacity of that material. And that very simple measurement gave a very surprising result. You see a graph here of a term named gamma. It was supposed to be 0 all along. It was not. And moreover, it even depended on the applied magnetic fields. So this unusual result led to two models trying to describe that data. Those two models really describe the data well, but have very different prediction regarding the magnetization of the material. So the next step is to measure its magnetization and try to differentiate if this material is really the quantum spin liquid I'm looking for. For more pretty graphs and some explanation about this quantum exotic phase, meet me at the poster next to the beginning.
All right. Thank you, Itai. Next, we have Nimrod Harpak. Uri Arieli, please come to the... Uri is here, okay. And Nimrod will tell us about large-scale self-catalyzed sponge-like silicon, right?
Okay, hi. I'm Nimrod. I'm from Fernando Patowski's group. Today I'm going to talk also a bit of something different, which is anodes for lithium-ion batteries. I will start off by saying that the current anode material that is employed in your batteries today, in your cell phones, in your laptops, is everything is graphite based. So graphite actually has very low capacities, and in order to progress into further applications such as electric vehicles, we need to find new materials. So silicon has been chosen by many around the world to be the next generation anode material, but it hasn't been commercialized yet since it has a lot of problems. So I will tell you a bit about what we did. We took an ordinary stainless steel and we treated it with hydrofluoric acid. The hydrofluoric acid actually is selectively etching the iron and alters the chemical species of the surface, which actually creates a catalytic island that further in the CVD can grow silicon nanostructures in very low temperatures. So what's special about this substrate is that it has an increased mechanical stability. And it doesn't delaminate from the surface because it has a lot of a short section of nickel silicide section that doesn't expand as much as the other elongated silicon structures. And what we did in our lab is actually it's very easy to upscale it, and we managed to do around 2 meters of the composite anode and employ it in a commercial battery. Which is currently being progressed. So if you are interested, please come to poster number 26. I'll tell you more.
Okay, so next we have Uri Arieli from Chaim Sochowski groups telling us about towards ultrafast phase spectroscopy. Rabi oscillation.
Yeah, it's okay. Hi, I'm Uri, and I'm a PhD student of Chaim Suchovsky, and I'll talk about ultrafast phase microscopy. So basically, when we're interested in processes like oscillations of electrons in metals or exciton dynamics in photosynthesis, they all occur on the picosecond to femtosecond timescales, and they cannot be resolved using regular electronics that work on the nanosecond timescale. So in order to address this, we've built an ultrafast phase microscope. It doesn't work. Okay. So to address this, we built an ultrafast phase microscope composed with the asymmetric FTL interference microscope and an 8 femtosecond laser. With this, we're able to resolve energy transfer between nanoparticles that happens within 10 femtoseconds, as well as nonlinear dynamics. If you're interested to hear more, I'm at poster 27. Thank you.
Wow. Uri did it in 1 minute, by the way. You did it in 1 minute. And you get extra credit for that, by the way. Next, we have Inbal Oz, coming from Avram Nitzan and Oded Hod groups, telling us when the edge rules. I would ask Dayanda to come. Oh.
Okay. So hi everyone, my name is Inbal and my poster is titled When the Edge Rules, Interlayer Transport in Graphene Dominated by Edge States. So we start by considering interesting experimental results that suggest when you look at a graphene flake, some atoms contribute more to the interlayer transport than others. So the experiment takes one graphene flake, places it on top of another one, and applies a bias voltage between them. As a result, electrons start traveling from the highest potential to the lowest potential, thus effectively creating an electrical current. But when you look at the graphene flake, some atoms, which are colored here in red, sit on the edge of the flake. The rest, a large number of atoms, constitutes the bulk. And it turns out that although the number of particles in the edge is smaller, for some reason, the current from these atoms is larger. To understand why, we treated graphene for what it truly was, which is a lattice, and we show that the way that you cut the lattice determines whether your wave function would be a bulk state, localized in the bulk, or an edge state, localized on the edge atoms. And based on this understanding, we derived numerical simulations that in turn enabled us to reproduce the experimental results. So for example, I can simulate two graphene flakes sliding relative to each other. I can plot the current as a function of the distance, and note how nicely the numerical results here in red reproduce the experimental behavior. Thank you.
Thank you, Inbal. Also ahead of time. I lost my, where's my list? I lost my list. Oh, here it is. Okay. So, next, we have Dayananda Desagani. I got it right? Okay, somehow. From Yossi Shaham groups, telling us about gold nanoparticle-based bioelectrochemical sensors. I do ask Mario to come and sit here.
Good evening, everyone. I am Dayananda from Yossi Shaham group. Today I am going to present about the So nanoparticle-based bioelectrochemical sensors, which can be used to plant as a Internet of Plants. Like means we can use the plant as a sensor. Suppose normally the commercial sensors which are present today is like if you want to check the water content of the plant, just we will put a sensors in the soil and check how much is the water content for the plant. If it is less, then we'll add the water. Or if in the same case for the nutrients also. or if anything, we'll check around the plant, not from the plant. So in this case, we are using the plant itself as a sensor. So for that, what we will do is, we'll connect a sensor like a chip, which is made with the gold, this one. So we'll connect to the plant, and we'll inject some substrate which is relevant to the enzyme present in the leaf. So here I have taken the case is a tobacco plant. The enzyme is the beta-glucuronide. So I have injected the substrate, the phenolphthalein beta-glucuronide, so that it will react with the enzyme and it will give the product. And the product can be analyzed using the chip which we are connecting to the plant. It's a plant. So this will give the, if you do oxidize of this product, we can get the results through this by chronopotentiometry, chronoamperometry. So if you see here this one, this is the blank PBS we have taken here. We didn't give any signal, but when you take in the substrate, it will react with the enzyme present in the plant and will give the product. So you can, if you want more details, poster number 32. Thank you.
32 for more information. Okay. Dana, come to the chair. Next we have Mario Urso telling us about low-cost synthesis of nickel-based nanostructures for sensing.
Hello everybody, I'm Mario Urso, a PhD student from the University of Catania in Italy, and I'm a visiting student here. And my work is on the low-cost synthesis of nickel-based nanostructures, which we then applied for sensing and energy storage applications. Well, nickel-based nanostructures are a promising material for this kind of applications, but typically they are produced by very expensive approaches. In this case, we developed a nice and low-cost approach to fabricate this kind of nanoporous structures, which is completely metallic, by doing a simple chemical buff deposition of nickel hydroxide nanowalls, and then by transforming them into these bunches of nickel nanoparticles by a simple annealing and reducing atmosphere. These nanoparticles allowed us to detect via non-enzymatic way glucose in those concentrations which are of interest to detect glucose in human saliva instead of blood, so a less invasive way. But we can also modify the surface of the nickel nanoparticles by transforming them into nickel oxide nanoparticles, and in this case we were able to detect nitrogen oxide at room temperature, and we can also realize some nice core shell architectures. In this case we take advantage from the thin nickel hydroxide shell and from the conductive nickel core to get higher specific capacity and thus high rate performance for battery and in general energy storage application. For other information, come to my poster, which is number 33, and it's for some reason the smallest one, so you can't miss it. Thank you.
Thank you, Mario. Thank you so much. Okay, so next we have Dana Cohen coming from Lihi's group and Yossi Shaham's group, telling us about another self-assembled structures. Next will be Irena. Where is Irena?
Okay. So, hi, I'm Dana. I'm from Lee Adler Abramovich Group in a collaboration with Professor Yossi Shaham, and I will present you my work about structural characterization of self-assembled superhydrogel. Self-assembled peptide nanostructure are increasingly exploited as functional material in biomedicine, nanotechnology, and fabrication of advanced materials. Those self-assembled peptides are great material that can form 3D hydrogel that can be used in the cell culture and also as drug delivery platform. Those materials have special and unique characterization, and I will show you, for example, the from co-assembly of FMOC diphenylalanine and FMOC pentafluorophenylalanine, we get a synergistic effect of the mechanical properties of the hybrid hydrogel. In my work, I chose to focus on FMOC pentafluorophenylalanine, that is also known for its antibacterial properties. I used the solvent switch method to create the hydrogel and tried to play with different solvents like DMSO and ethanol, and I also played with the concentration of the peptide. With microfluidic system, I could see the self-assembled process and get this very cool video that showed me I will show it again. You can see we get different morphologies, spheres, fibrils, and tubes. I also did SEM images and XRD powder diffraction because one of my conditions create crystal. For more information, come check my poster, number 34.
Thank you so much, Dana. Next is Irena Grigoriant. Oh, wow, that's easy. Yeah, from Lee groups as well. Where is yours?
Hi, I'm Irena from Lee Adler Abramovich Group, and I would like to present the poster named Peptide Self-Assembled Nanostructures for Enzyme Encapsulation. As we know, the enzymes are extremely efficient biocatalysts, but their approach are limited because of few problems such as short shelf life and low reusability and thermal stability. So to resolve some of these problems, we are using a new approach. This approach for this approach, we are encapsulating enzymes and deliver them by using self-assembly building blocks to form peptide-based nanostructures, nanospheres from the left side, on the left side, and hydrogen nanoparticles on the right image. And both of them can act as enzyme nanocarriers. The enzyme we choose is able to destroy the cell-to-cell communication in bacteria that cause plant diseases. Okay, encapsulated enzyme maintain its activity with a slower and prolonged kinetic in the blue line compared to the unencapsulated free enzyme in red line in both nanocarriers. In addition, hydrogen nanoparticles with encapsulated enzyme show a high negative zeta potential that indicate electrically stable colloidal system. So you are invited to visit my poster.
All right, thank you. Last and not least, we have Tal Ma'aravi, coming from Oded Hod's group.
Okay. Okay. Hi everyone, my name is Tal and I'm from Professor Oded Hod's group, and my poster is titled Simulating Electron Dynamics in Open Quantum Systems Under Magnetic Fields. So the field of molecular electronics aims to use single molecules as electronic components. A typical molecular electronic device consists of a molecule that is coupled to two leads. A bias voltage is applied in the system, and therefore electrons flow from the source to the drain, thus generating a current in the system. Okay, so one of the major challenges in the field is to understand how to control the electron transport properties in such molecular junctions. One way to do that is to exploit interference phenomena in molecular rings. And the way one way to manipulate this interference effects is by applying a magnetic field, external magnetic field, and manipulate it via the Aharonov-Bohm effect. So we developed a theoretical method to simulate and study these effects in open quantum systems. We demonstrated our method on two simplistic hydrogen rings, which are different in what angle they are connected to the leads. And we analyzed the current transient dynamics upon application of a magnetic field. And if you want to hear some more details, please come see me at poster 31.
Thank you, and thank you all for a lovely day.